Hello students. In the previous class, we discussed about prokaryotic cell. In that, we completed cell envelope and genophore. So we'll continue the session. Now, apart from cell envelope and genophore, a bacteria or else a prokaryotic cell, they may consist of pigmented photosynthetic structures. And these structures, they are called as chromatophores. Chromatophores. And these chromatophores, as they are pigmented, they can help in photosynthesis. And these are particularly observed in photosynthetic bacteria like blue-green algal members. Now, apart from these structures, a bacteria consists of surface appendages or as extensions you can say. Now, these extensions, they may help in conjugation or attachment, or it also helps in providing motility to the cell. Now these extensions, they can be tubular extensions. So they can be tubular. If they are tubular, they are called as pili. They can be hair-like, bristle-like extensions called as fimbriae. Or else it can be a thread-like extensions. Now these are flagella, which helps in motility. Now these three appendages, they are made of different kind of proteins. Pili, that is made of pillin protein. Fimbriae, that is made of fimbrilin. Flagella, that is made of flagellin protein. Now this pili, it is tubular in structure that helps in conjugation or else mating. Number 2, fimbriae. They are hair-like or bristle-like we said, or fiber-like structures. They help in attachment. They help in attachment to the host body or else to the substratum. Next, flagella. Now this is a structure that helps in providing motility, and this helps in motility here. It means locomotion. Now this is a filament-like or else it is a thread-like, filamentous, long structure that helps in motility or locomotion. Now in all these three things, flagella, a little more detail we have to observe. So if we talk about flagella, it consists of three parts basically. It consists of a basal part, and here if you talk about this basal part, it is called as basal body, and this basal body, they may consist of 2 disc or else 4 disc-like structures that can be present. So here it consists of a basal body. So here this is a basal body. Basal body. And this one is connected to the muzzle piece. So here there is a muzzle piece or hook, and over the top it consists of a filament. Now the basal body, it consists of 4 discs, and these 4 discs, they are embedded at different regions. Now here the basal disc is called as M disc. Now that is attached to the base of plasma membrane. Number 2, the above one is a S disc that is attached to the surface of plasma membrane. Number 3, that is called as P disc, that is attached to peptidoglycan layer. Peptidoglycan. And coming to the last, that is L disc, which is attached to LPS layer. I already mentioned in the previous session, in the previous video, LPS layer, that is a characteristic feature of gram-negative bacteria. Now here, how many discs are observed in gram-positive bacteria? So here, 2 discs are observed in gram-positive. When it comes to gram-negative, all the 4 discs are present in the flagella. So this is all about the surface appendages. Now apart from these things, it also consists of non-membrane bound structures also. Now these non-membrane bound structures, that includes number 5, ribosomes. Ribosomes. Now these ribosomes, they help in protein synthesis. They are attached to the plasma membrane. They range between 15 nanometer to 20 nanometer in diameter. And the type of ribosomes which are observed in prokaryote, they are 70S type of ribosomes. And these ribosomes, they are made of two subunits. The larger subunit is 50S subunit. The smaller subunit is a 30S subunit. And unitedly, it makes the 70S subunit of ribosome, or else 70S type of ribosome. Now, what is the function of ribosomes? They help in protein synthesis. Now here, during the time of protein synthesis, they are observed in attachment to mRNA. So many ribosomes, they are attached to one mRNA or messenger RNA. And this combination, so here I'm taking the structure like this. So one mRNA, many ribosomes. This is called as polyribosome or polysome, or else it is also called as ergosome. Now this polyribosome is the one which helps in converting the mRNA, translating the mRNA into the protein. Now apart from all these, the last one, that is number 6, it also consists of inclusion bodies. Now these inclusion bodies, they are non-membrane bound. So you can get different kind of questions based on these all aspects individually as well. So here it's a non-membrane bound. It's a non-membrane bound. It consists of reserved materials, dead materials, right? So it helps in storage, you can say. And where it is present freely in cytoplasm. Freely in the cytoplasm, that will be present. Now, what are those different types of inclusion bodies that are present? It includes glycogen granules, glycogen phosphate, synophysin granules that are present. So if a question is given, out of the following which is odd with respect to inclusion bodies? So how you have to take the question? Number 1, glycogen granules. Number 2, phosphate granules. Number 3, synophysin granules. And number 4, sap vacuole. So what is a suitable answer? Sap vacuole you can take as a suitable answer. Now apart from all these, okay, there is a small, right, special case is present. It consists of, in the prokaryotes, they consist of gas vacuole. It is also a non-membrane bound vacuole. It's an air-filled space which helps in buoyancy. And where these are particularly present, buoyancy, that means that helps in floating. Now where these gas vacuoles are present, it is a characteristic feature of prokaryotes. For example, like blue-green algae and green photosynthetic bacteria. Green photosynthetic bacteria that consist of the gas vacuole. So this is all about the prokaryotic cell. So let's try to solve some questions. Identify the wrong statement for inclusion bodies. Inclusion bodies, they include reserved materials. They are freely present in the cytoplasm of prokaryotes. They are non-membrane bound. They also include certain air-filled spaces called as gas vacuole. So here, they lie freely in cytoplasm. That's true. They store reserved food materials. That's true. They are not surrounded by any membrane. Yes, that's also true. They provide buoyancy only. And do remember this point, many a times, only, all, these terms, if it is given, most probably such kind of statements can become wrong. Number 11. Select the option with correct sequence of cell envelope layers from outside to inside in a prokaryotic cell. From outside to inside. So outside what is present? Glycocalyx followed by cell wall followed by cell membrane or else plasma membrane. So what is that which is suitable here? That is C, which is suitable. Number 12. Which of the given cells is round and oval in shape? In the overview we have studied about it. What is that which is round and oval in shape? Mesophyll cells. Question number 13. Which is a non-membrane bound organelle found in both prokaryotes and eukaryotes? Non-membrane bound organelle, it can be ribosomes and centrioles, but it is observed in both prokaryotes and eukaryotes has been given. So the suitable answer, that is 70S type of ribosome. Don't get confused. 70S is observed in prokaryotes as well as in cell organelles like chloroplast and mitochondria of eukaryotes. Question number 14. Prokaryotes do not have cell membrane. No, that is present. Cell wall. Yes, that's also present. Nucleus. So here number C, which is the suitable answer. Gas vacuole are found in all except. It is observed in blue-green algae, purple and green photosynthetic bacteria, not in mycoplasma. Which of the following cell organelle is absent in higher plant cells but present in animal cells? So this is also from the overview which we have discussed. The non-membrane bound cell organelle which is absent in plant but present in animal cell, that is centriole. Number 17. Select the incorrect statement. Bacterial cells may be motile or non-motile. Yes, that's true. The villi are elongated tubular structures made up of special protein. Yes, it is made of pillin protein. Gram-positive bacteria do not take gram stain. Okay, mesosomes help in DNA replication. So here C is the suitable answer. As it takes the gram staining, it is giving a positive effect to the gram staining process, so-called gram-positive bacteria. Let's solve some previous year questions from the topic of prokaryotic cell. Number 18. Inclusion bodies of blue-green algae, purple and green photosynthetic bacteria are. So what is that inclusion body which is present in these three? Already we have completed similar kind of question earlier also. That is gas vacuole. Number 19. Cell wall is absent in. So cell wall is absent in mycoplasma, which is a wall-less moneron or else wall-less prokaryotic cell. Number 20. Which structure perform the function of mitochondria in bacteria? So who is performing the function of mitochondria in bacteria? So here first of all, mitochondria function is what? It helps in performing ETS, respiration like aerobic respiration, helping in production of cellular energy like ATP, and that function will be done by one of the structure in bacteria, that is mesosome. So this is all about the questions of prokaryotes. In the next session, we'll go with eukaryotic cell discussion. Thank you.